Hello, everyone. My name is Ahmed. I'm a third year pathology resident, and today I'm going to be talking about the differentiation of hematopoietic stem cells to different cell types and the associated markers that are expressed on these cells and the implication on classifying acute leukemia. So let's get started. So, first of all, hematopoietic stem cells can give rise to multipotent progenitor cells, and multipotent progenitor cells can give rise to, or they can differentiate into, common myeloid progenitors and common lymphoid progenitors. Common myeloid progenitors can further differentiate into either megakaryocytic erythroid progenitors or to granulocytic monocytic progenitors. Megakaryocytic erythroid progenitors, as the name suggests, can differentiate into megakaryocytes or to erythroblasts. Megakaryocytes can differentiate to megakaryocytes, and megakaryocytes can give rise to platelets. Erythroblasts can differentiate and give rise to mature erythrocytes or mature red blood cells. Granulocyte monocyte progenitors, on the other side, can give rise to myeloblasts or to monoblasts. Myeloblasts can go through different stages of maturation, differentiating into promyelocytes, myelocytes, metamyelocytes, and eventually giving rising to neutrophils. But they can also differentiate into eosinophils as well as basophils. Monoblasts can differentiate into promonocytes and other forms and eventually give rise to monocytes. And monocytes can give rise to macrophages. Common lymphoid progenitors, on the other hand, can give rise to B lymphoid progenitors and T NK progenitors. B lymphoid progenitors can give rise to mature B cells, and T NK progenitors will give rise to T lymphoblasts or T lymphoid progenitors, and NK progenitors. T progenitors are going to give rise to mature T lymphocytes or mature T cells, and NK progenitors are going to give rise to NK cells. As for the markers that are expressed on these cells in this flowchart, hematopoietic stem cells are CD34 positive. Let me use a different color. Are CD34 positive. Multipotent progenitors can differentiate into common myeloid progenitors as well as common lymphoid progenitor. Both myeloid and lymphoid progenitors are going to be dim CD45. They're going to be CD34 positive. CD38 can be positive, and HLA-DR can be positive as well. As for common myeloid progenitors, they can be positive for CD123. But CD123 is not a specific marker, and it can be expressed on different cell types, including basophils and plasmacytoid dendritic cells. Both basophils and plasmacytoid dendritic cells tend to have bright CD123 expression, and the way by which you can differentiate these two cell types from each other is by looking at HLA-DR. Basophils are HLA-DR negative, and plasmacytoid dendritic cells are HLA-DR positive. As for megakaryoplasts, these tend to be CD34 positive, and they show variable expression of CD41, CD61, and CD42B. And as they mature, they show more expression of these megakaryocytic markers, and platelets express these markers as well. As for the erythroid progenitors, these erythroid progenitors show expression of CD71. CD71 is a pan-erythroid marker, however, it's not really specific. CD117 tend to be positive in the immature erythroid progenitors, and as these cells mature, they lose expression of CD117, and they gain expression of glycophorin. The other name for glycophorin is CD235A, and they can show expression of hemoglobin as well. Now, let's go for the granulocyte monocyte progenitor. These cells, the myeloblasts, are CD117 positive. This is a marker of immaturity. There are other markers that are expressed throughout myeloid differentiation. These include CD13, CD33, CD15, CD65, CD11B, CD16, and myeloperoxidase. Myeloperoxidase is the most specific myeloid lineage marker. Monoblasts, on the other hand, are CD117 negative. They are MPO negative as well. They share common antigens with myeloblasts or with myeloid cells, including CD13, CD33, and CD11B. However, they can express monocyte-specific antigens, including CD11C, CD14, and CD64. And generally speaking, CD14 expression tend to be dim in immature monocytic precursors and tend to be more expressed in mature monocytic precursors, in more mature monocytes. Now, when we go to the common lymphoid progenitors, these can be dim CD45, they are CD34 positive, CD38 positive, HLA-DR can be positive in these. A common marker that is positive in both B lymphoid as well as T lymphoid progenitors is TDT. So TDT is a marker of immaturity. Another marker of immaturity in B lymphoblasts is CD10. And for T lymphoblasts, a common marker of immaturity is CD1A. As these B lymphoblasts mature into mature B cells, they lose expression of CD10 and they gain expression of other markers like CD20 and CD22. CD19 is expressed throughout B cell maturation, and it's a highly sensitive and specific marker for B cell differentiation. Cytoplasmic CD3 is a common marker throughout T cell maturation. Another common marker in T cells is CD7. And then CD2, CD5, CD4, and CD8 can be variably expressed. But the most specific marker for T cell differentiation is cytoplasmic CD3. As for NK cells, these tend to be CD16 positive and CD56 positive. Now that we understand the general immunophenotype for these cells, let's discuss how we're going to use that for a classification of acute leukemia. So if you have a case of acute leukemia, common immature hematopoietic markers in general are DIM CD45 as well as CD34 positivity. However, remember that not all blasts are CD34 positive. Now, when we talk about myeloid, you can have myeloid, acute myeloid leukemia, or you can have lymphoblastic leukemia lymphoma, which can be B lymphoblastic leukemia lymphoma or T lymphoblastic leukemia lymphoma. For myeloids, acute myeloid leukemia, you want to see a population that is DIM CD45, CD34 positivity is good. A marker of immaturity is CD117. Then you want to see markers of myeloid differentiation. These markers of myeloid differentiation that can be seen in more mature forms of myeloid cells include MPO, myeloperoxidase. This is the most specific marker for myeloid differentiation. Other good markers are CD13, CD33, CD15, CD65, CD11B, and CD16. When you talk about monocytic leukemia, monocytic leukemia is MPO negative. They can show common myeloid antigens like CD13, CD33, CD11B, but more specific monocytic antigens include CD11C, CD14, and CD64. B lymphoblastic leukemia lymphoma, on the other hand, you can see DIM CD45, CD34 positive population. Markers of immaturity include CD10, CD38, and TDT. Then you need markers of B lymphoid differentiation. We said that CD19 is expressed throughout B cell maturation. And what you're looking for here is strong CD19 expression. Other markers of B cell differentiation that you can see include CD20, CD22, and CD79A. But CD79A can also be expressed in T lymphoblastic leukemia lymphoma. So if you don't have CD19 expression or CD20 expression, you should not consider CD79A as a marker of expression of B lymphoblastic leukemia lymphoma in your case. As for T lymphoid, these can be DIM CD45, CD34 positive, CD38 can be positive. TDT is a good marker of immaturity. Another good marker of immaturity is CD1A. A marker that is present throughout T cell maturation is cytoplasmic CD3. And this is the most specific marker for T cell differentiation. Most cases of T lymphoblastic leukemia lymphoma show CD7 expression as well, so CD7 is a very good marker. Additional T cell markers are helpful, like CD2, CD5, CD4, and CD8. Now, to make a diagnosis of acute myeloid leukemia, you want to see at least 2 myeloid-associated antigens, one of these antigens, and then MPO positivity is good for making a diagnosis of acute myeloid leukemia. However, acute myeloid leukemia can show lymphoid markers expression, and lymphoblastic leukemia lymphoma can show acute myeloid leukemia markers expression as well. So it's not about finding a specific marker, it's more about the big picture If you see a couple markers of myeloid differentiation, even if MPO is dim, you can go ahead and call it an acute myeloid leukemia. If it doesn't satisfy criteria for another leukemia, like a mixed phenotype acute leukemia, for example. For B lymphoid, you want to see CD19 expression. If you have strong CD19 expression, that's actually very good. But CD19 can be expressed in acute myeloid leukemia. So when you have multiple markers like CD19, CD20, CD79A, for example, then you can confidently make a diagnosis of a B lymphoblastic leukemia lymphoma. As for T lymphoid, you're looking for cytoplasmic CD3 expression. But then you want to see more markers like CD7, CD2, CD5 positivity to make the diagnosis. About less than 4% of acute leukemia cases fall under the category of mixed phenotype acute leukemia. Mixed phenotype acute leukemia is when you have immunophenotypic evidence of more than one leukemia differentiation. You can have two blast populations, like for example, one blast population with one phenotype, and then second blast population with second phenotype. That can be mixed phenotype acute leukemia, but you can also have one blast population with two phenotypes showing immunophenotypic evidence of myeloid and B lymphoid differentiation, for example. You can have myeloid and B lymphoid, you can have myeloid and T lymphoid, and you can have a combination of myeloid, B lymphoid, and T lymphoid, and you can have other rare combinations. To make a diagnosis of mixed phenotype acute leukemia, there are strict criteria that you need to follow, and then we can discuss that in a separate video. As for now, just remember, it's not about finding a specific marker. It's all about the overall picture, taking into account the morphology, the immunophenotype, as well as the genetic findings. This is the end of this video, and see you next time.